In today's video, we're going to talk a little bit more about the sliding filament theory um, with muscle contraction. So I kind of went over that yesterday. So just wanted to kind of start here. And so basically what's going on at a microscopic level, the Z lines within our sarcomere are each going to be moving closer towards each other, hence the sliding part. And actually, the proteins that are doing the sliding are the actin filaments. And so as you look at subsequent diagrams here, when we're having a muscle contraction, those Z lines get closer together, and that's because those actin proteins are being pulled or slid over the myosin proteins as the contraction takes place. So a reminder again, we got cross bridge cycling. The cross bridge heads on our myosin are able to go up and make contact with the actin active sites. Remember, that only happens if calcium's present within our muscle fiber. So the calcium has to be released from the sarcoplasmic reticulum, and then that myosin cross bridge head is able to make contact with that actin site. And so you can see the calcium has attached to the troponin complex. That troponin complex, remember, is also attached to the tropomyosin, so it now slides out of the way, exposing that active site on the actin. So now we're going to use ATP to get the cross bridge head to sort of ratchet, if you will. So the myosin head makes contact with an active site, and you can see that as we step from here to here, we've made contact. And so we're going to use the ADP, and phosphate actually gets released as we go from this picture to this picture, released from that myosin head. And so now the myosin head actually kind of ratchets forward, sliding that actin filament a little bit. So more ATP now reattaches to that myosin head and binds to it again. And so that ATP is going to reset, or cock, if you will, that myosin cross bridge head into place, cock it into place. And so now it can reattach, and we start the whole cycle over and over. Now keep in mind, in this picture we're only showing a few of these myosin heads. They're not all working at the same time when they go up and attach. For, let's say there's 10 cross bridge hedges that have gone up and made contact with the active site. There's 10 that are in the process of sort of being reset, if you will, back to their original process. And you'll see that in one of these slides coming up here. So just so you kind of get an idea of what's going on, we'll walk you through. Here you can see a diagram of the sarcomere. Our Z lines are heavier myosin filaments, and then the actin are in more of a yellow there. Okay. So if we go to the next slide and zoom in here a little bit, you can see our Z line, our myosin, our actin, our troponin tropomyosin complex there. And so right now our cross bridge head is not able to go up and make contact with the active site because there's no calcium. So we'll add some calcium. The calcium is going to come down, attach to the troponin. The troponin tropomyosin complex moves out of the way, and now that cross bridge head can make contact or bind with that active site on the actin. Okay, so the ADP and phosphate leave, and we ratchet forward. That actin begins to slide. Now the same thing's going on the other side of the sarcomere as well. Okay, so now we need some ATP to come in, and that cross bridge head is going to detach. Okay, we're going to separate that ATP into ADP plus phosphate again. And so now we're going to take that energy, and it's going to reattach to the next active site, sliding it forward. And then that process just continues until we've reached the contraction. As you see there, the calcium just left. So now there's no calcium. The cross bridge head can't make contact, so the actin slides back to its resting spot. We want to contract again. The calcium comes up, binds to the troponin. The tropomyosin moves out of the way. The cross-bridge head makes contact with the active site. ATP is used, and we can detach, reset, connect to a new active site, and continue that process anytime we want to have a contraction. So we got a process of contraction and sort of relaxation going on here. Now, if I can get to the next slide, we'll be good. So here's a kind of a breakdown, a little animation for you to watch that'll kind of give you a similar situation.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ATP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
So the question now becomes, how does relaxation actually take place? If you remember in the previous video, when the neuron was releasing acetylcholine, one of the actions was to break down that acetylcholine with an enzyme called acetylcholine esterase. So the enzyme actually breaks down the acetylcholine, into its choline part and its acetic acid part. And so as we break those down, now there's no acetylcholine in that synaptic cleft to open those sodium gates to have sodium rush in and create electrical signal. So once all the acetylcholine's gone, there's no sodium rushing in to generate a new electrical signal. There's no electrical signal to go throughout the sarcolemma, including down the transverse tubule. If there's no electrical signal going down the transverse tubule, no more calcium is going to be released. So the muscle electrical impulse stops because there's no acetylcholine esterase. There's no stimulus going down the sarcolemma throughout the muscle fiber. So now the calcium's actually going to move back into the sarcoplasmic reticulum or cisternae. Well, if there's no calcium, the calcium isn't going to attach to the troponin, so that gets removed. So now that troponin tropomyosin complex cover up that active site, and now the myosin is that cross bridge head is prevented from making contact. So that muscle fiber actually goes back to the relaxed state, and the sarcomere goes back to its normal position in order to contract.